related to the third goal of Sustainable Development Goals 2030, especially to eliminate of neglected tropical disease such as dengue hemorrhagic fever. Based on records from the World Health Organization, an estimated 500,000 DHF patients need hospital treatment each year. This disease is included in the 10 causes of care in hospitals and other health facilities and death in children in at least 8 tropical countries in Asia. Various efforts have been made in anticipating an outbreak, start from using the anti-mosquito spray to lotion. However, research show that anti-mosquito drugs are not entirely beneficial. The use of chemical mosquito repellent can leave the residue, pollute the environment, and can cause the resistance. Residual residue can cause health problems such as irritation, muscle spasms, disrupt the respiratory system, and even triggers the occurrence of asthma and cancer. Therefore, concrete efforts that are safe and effective for humans and the environment are needed. One of them is by applying an idea from me, Refa Rahmadiansyah from Indonesia, entitled BAKULIS, Airspray solution from Jeruk Limau peel waste and lemongrass as natural larvicide for anticipating dengue hemorrhagic fever. Hemorrhagic fever can make a patient's body's temperature very high, usually accompanied by headaches, joint muscles and bones, and pain in the back of the eye. This disease is caused by dengue virus which spread through the bite of the Aedes aegypti mosquito. The mosquito breeds a lot in densely populated areas with humid and warm climates. This research is purely experimental research which possesses only control group design by making airspray solution products from Jeruk Limau peel waste and lemongrass, which are analyzed descriptively and analytically. Experiments carried out with randomized complete design 3-time repetition to analyze the effectiveness of this product. The study used 2 groups, experimental group or P using the products and control group or K using the aqueous in the culture container. Besides, we use the hedonic organoleptic test for measuring the public preference of this product by questionnaire. And we did the observation of the larval death and acute effects or any kind of allergic reactions caused by BAKULIS. This research was started from the collecting the ingredients and tools, ingredients weighing, invalidation method, and extracting process until it's done as ready-made product of BAKULIS. Then, we did the observation of the larval death and allergic reactions. And last, hedonic organoleptic test. And here are the preference of the respondents that based on hedonic organoleptic test, all the people like this product. For testing the toxicity, there were no reports of toxicity from respondents either from the allergic reactions or any acute effects. So what contains in Jeruk Limau and lemongrass? Previous research stated that the results of the phytochemical tests of the lime samples from field extract and all gave positive results from the content of alkaloid, tannins, saponin, and flavonoids. As for lemongrass extract, it gave positive test results from the content of saponin, tannin, alkaloid, quinones, and steroids. This content is thought to kill the Aedes aegypti larvae by inhibiting the inhibition of the acetylcholinesterase enzyme mechanism on the nerve impulses and reducing their ability to digest food. These bioactive compounds is cause the larval death because it is unable to detoxify these toxic compounds that enter the larva's body. The mechanism of action of larvicides in killing larvae such in 3 ways, such as contact poisoning through the body wall of the larvae through contact with the skin, penetrating the cuticle, trachea, or sensory glands or other organs related to the cuticle. The body wall of insects is the part of the insect's body that can absorb large amounts of toxic substances through stomach poisoning by mouth and as a respiratory poison or fumigen. So here are the SWOT analysis of this product. Here are the strengths, the weaknesses, the opportunities, and the threats. So we can conclude that this solution was proven to be effective as a natural larvicide against the manufacture of dengue hemorrhagic fever, the Aedes aegypti mosquito. As for the results of the Vaver level test, satisfactory results were obtained where all the respondents liked the product and has a potential to produce massively. It is suggested for the further research to determine the optimal dose by increasing the number of control groups from various concentration and increase the sample size so it can be commercialized as a safe and effective larvicide product. Here are the references that I use. Dear ladies and gentlemen, that's all my idea and innovation. Hope that you can interest and it can be applied to solve the problems. Save your hope and explore the nature. Thank you for attention and thank you for the chance. Goodbye.